Hi, my name is Scott,
I'm Kristen.
and we're going to go over the group problems for lesson 12. Um, and these ones were mostly over, um, it was over the lac operon. Yeah, thank you. Lac operon and a little bit about operons in general, but mostly the lac operon. The first question asks a question about certain things that are needed for the lac operon. And it gives us some terms here and asks us which one is compatible with which. So the first one is about regulatory proteins. And so the things that are needed for regulatory the terms that are an example of like regulatory proteins are the repressor, the activator, and cap. And so those three are the three regulatory proteins. B asks about an example of a small effector molecule. We said inducer, allolactase, and CAMP, or CAMP, cyclic AMP. For C, it says which term describes a sequence that is found in DNA. So all the sequences that were found in DNA that are on this list is +1 and -10, which we learned about earlier in transcription. And then there's the LAC-A and the LAC-Z that are both structural genes on the lac operon. D, it says which one is not involved in the lac operon. The answer is none of them. All of them are needed in some way, shape, or form for the lac operon. And E says which of the above is a structural gene, and the two structural genes were LAC-A and LAC-Z. Those are two structural genes that are needed for the lac operon.
Okay. And then, so for question number 2, it's just fill in the blanks. So for part A, it says the lac operon is operated by 2 inducers, and those 2 are going to be allolactase, lactose, and CAMP, cyclic AMP. And then B, it says the binding site for the sigma factor and RNA polymerase is called the lac promoter, while the cap site is the binding site for the activator that regulates the lac operon. For C, the enzyme that converts ATP to CAMP is going to be adenyl cyclase. For D, the -35, -10, and +1 sequences are found within the promoter or LAC-P portion of the lac operon. For E, the LAC-L gene encodes the lac repressor, while the LAC-Y gene encodes lactose permease. And then for part F, it asks what can regulate gene expression in bacteria by causing transcription to terminate prematurely. That's going to be attenuation. And then for part G, an E. coli strain that contains 2 copies of the lac operon is called a merozygote strain.
So question 3, and I'll show my paper to show how, or just the answers that I got. So the first one here is where IPTG is present, glucose. And so I said it's going to be non-blue because IPTG is needed to release the repressor from the operator. Question letter B, the IPTG is present, the glucose is absent, and XGAL is present. And so this one is actually, oh, sorry, I read the wrong one. ITPG is present, glucose is absent, XGAL is present, and so that one is going to be blue because all the necessary parts are needed. For C, IPTG is present, glucose is absent, and XGAL is present, and so this is non-blue. It has everything that it needs except for XGAL, which turns the cells to a blue color. And then IPTG is present, glucose is present, and XGAL is present. So this one can't work because we need cyclic AMP, which will not be produced if glucose is present. And then the last one is IPTG is present, glucose is present, and XGAL is absent. So this one can't work because the presence of glucose stops cyclic AMP from being made, and also XGAL is needed to turn the cell blue. So that's how we got the questions to that one.
Okay, for question 4, it gives you just different genes on the lac operon, and it asks if these E. coli strains display constitutive, inducible, or non-inducible expression of the LAC-Z gene. So for part A, I guess it has, oh gosh, I don't know if you, sorry, my handwriting's really bad. I, P, O, and Z, and they all have pluses on them. And so for that one, it's going to be inducible because that LAC-I will be able to stop the transcription. For part B, it's going to be IS, and then P plus, O plus, and Z plus. And that one is going to be non-inducible because it's just always going to be stuck there and not ever bind. And then for part C, it has this I plus, P minus, OC, Z plus for the chromosome, and then I plus, P plus, OC, and Z plus for the plasmid. And so C is going to be inducible. D is going to have I plus, P plus, O plus, and Z plus for the chromosome, and then IS, P plus, O plus, and Z plus for the plasmid. And that one is going to be non-inducible because the plasmid will create a repressor and it'll affect both. And then for the last one, it's going to be IS, P plus, O plus, Z plus for the chromosome, and then I plus, P plus, OC, and then Z plus for the plasmid, which is going to be constitutive.
Cool. Now we're into the last one here, question 5, and has questions about an E. coli genotype. It gives the E. coli genotype, and then asks if lactose will turn, or if the cell will turn yellow, or if lactose will turn yellow or not. And so I have it written here. And on this first one, it gives us this gene. And for us, we said it was 0. And the reason why is because of this. The promoter site has a minus on it. And so that will actually cause nothing can be made because of that. For B, we said it's 1-1. And that is because of this little guy right here. And that will actually cause this is for the operator, O is for the operator, and it's a defective operator sequence, and the LAC repressor can't bind to OC, so we'll only have 1. And then for C, we have this one is normal, this top one, but then the bottom one has the same issue here, where OC, and so for the first one, 2, and then for the second one, just 1. For D, we have, let's see, so we have this one, it's making, because of the I here being defective, it will cause this one to, what is it called? It will continually create more and more of this enzyme. However, because of this P site on the other one, there will only be 1 and 1 made. And then for the last one, there's IS here. And so this will actually create a repressor that can bind to the operator but can't bind to allolactose. And that'll be enough, it'll actually create enough of it that it'll bind to both of them. And so there will be nothing made.
Okay, thanks for watching, guys.
Mm-hmm.